Hi, my name is Sarah Matthews, and I'm from the Poche Lab at McGill University, and I'll be presenting the work we did in collaboration with l'Université de Polytechnique, which was to determine the diversity of sequence types that can be captured with Legiolert, as well as test storage methods for that assay. So, culturing on standard media has long been the gold standard for Legionella detection and quantification. However, it requires multiple manipulation steps and can take upwards of 10 days to get fully validated results. It is also quite sensitive to interfering flora. Legiolert is a rapid culture-based enzymatic assay with comparable sensitivity and selectivity and is much more streamlined and can take only 7 days to get fully validated results. Additional steps can be added to get colony isolation from valid plates where you simply streak the media onto GVPC, and then you can take these colonies and use them for characterization. We added an additional validation step just to be doubly sure that we were getting pure Legionella by replating colonies with and without cysteine and simply taking those that did not grow in the absence of cysteine. So using this procedure on real samples, we hope to determine the diversity of the STs Legiolert can capture, and then also tried several methods to preserve the media for later characterization. Starting with non-potable water results, we used Legiolert on a total of 165 and had only 5 invalid results. So that's those where the tray had an abnormal appearance or we found non-Legionella-like colonies. 5 represents a very low frequency of invalid results among non-potable water samples. As for the positive ones, we went forward with the isolation, and also for a subset of these samples, we also did standard culture method for comparison and found that for 6 out of the 7 where both methods were positives, the ST types matched, albeit just for ST1. And this is because the standard culture only was able to capture ST1 sequence types. In comparison, Legiolert was also able to detect 2 more STs, such as 3054 and 3069. So for potable water, we had 253 samples. There were no invalid results and a very high positivity rate at the 2 locations we sampled. One location was completely dominated by a single ST, ST378, while the other location had 2 ST types, ST1427 and ST2859. And there were actually 2 incidences where the 2 STs were found within the same packet. This prompted us to examine the interval variability of a highly positive packet by isolating colonies from 9 individual wells, 4 large and 5 small, in addition to a composite and performing whole genome sequencing. We found 2859 to be dominant, with only 1 well detecting from which we detected ST1427, and then analyzed the rest of the 2859 genomes and found that it was subspecies Frazeri, and also a SNP analysis separated them into 2 phylogenetic groups that differed by 44 SNPs. Isolates from small wells always clustered together, while those from large and composites split between the 2. This makes sense due to the larger size of large wells are more likely to be inoculated by more than 1 bacteria, and a composite is made from multiple wells. But this left the question of what size composite would be most representative. So to answer that question, we decided to model the sampling probabilities by combining a binomial distribution to represent colony sampling, and we can do this because of the large number of colonies on the plates. We could consider that the frequency of the strains remain the same between every trial, so between every colony you pick off the plate. And we used a hypergeometric distribution for well sampling since they are not being replaced every time you pick a well. So combining these two, we see that there are diminishing returns for increasing composite size, since while you're increasing your chance of choosing a well that has the minor strain, you're decreasing its proportion in the final composite, which makes it harder to find there. So there's not much benefit in going beyond 10 wells in your composite size. Okay, and lastly for the storage test. So storing the media from Legiolerts as they are at different temperatures did not really preserve viable bacteria for long, ending usually after 4 months. But the simple addition of glycerol can keep them alive for up to a year, even at -20°C, albeit to a lesser extent, and pretty much completely when you lower the temperature further to -80°C. And this was comparable to standard AYE and glycerol stocks that we use already in the lab to store Legionella isolates. So to summarize results, Legiolert is a streamlined alternative to standard culture that can detect the Fusarium subspecies as well as new and clinically relevant STs. A composite of 10 wells is representative of the tray based on WGS and modeling data, and glycerol can preserve samples for the long term, even at -20 or -80 degrees C. So we propose this procedure for storing Legiolert media, which contains a population of isolates, by adding glycerol, which can be frozen immediately and streaked out later, or can be streaked out immediately onto GBPC and produce colonies for characterization. Thank you.